Hello, my name is Nidia Truval, and I'm a researcher at the Fogarty International Center at NIH. I am a molecular epidemiologist of infectious diseases working with genetic data to investigate the different aspects of viral evolution, spread, and adaptation. In this section, we are going to introduce the concept of phylodynamics and how we can use it to reconstruct and infer evolutionary histories from genetic sequence data. This will provide the necessary background you will need to navigate the BEAST package in the tutorial sessions. Phylodynamics is defined as the study of how epidemiological, immunological, and evolutionary processes act and potentially interact to shape pathogens' phylogenies. On a historical perspective, phylodynamics merges two concepts. The first is the concept of modern epidemiology linked to its founding father, Dr. John Snow. In the mid-1800s, he used a map of London to illustrate how the cases of cholera centered around a public water pump. By removing the public water pump handle, he ended the cholera outbreak. Snow demonstrated through epidemiological studies that water could serve as a vehicle for transmitting cholera, and that epidemiological information could be used to direct prompt and appropriate public health action. The other concept in which phylodynamics is based on is evolution by trees, firstly conceptualized by Charles Darwin in the 1850s. The assumption here is that rapidly evolving pathogens accumulate mutations during the process of spread, and by sampling genetic data with these mutations through time, we can start to reconstruct this process. Although nowadays the sketch tree on the right is one of the most popular of Darwin, the only tree he published was the one on the left, explaining how species differentiate over time. Like Darwin studied the different phenotypes of differentiating species, we can study the genetic data of viruses because of their high evolutionary rate. Since patterns of spread leave an imprint on the viral genome, we can track the locations of our samples to study how a virus spreads. As John Snow, we live in times of dangerous epidemics, particularly those caused by fast-evolving RNA viruses that accumulate mutations during the process of spread, also known as measurably evolving populations. This kind of ancestral reconstruction has been used extensively in molecular epidemiology to uncover the phylogeographic dynamics of pathogens and to test hypotheses on the factors that drive epidemics, such as distance, air travel flux, among others.